Good afternoon to one and all. The presentation is enhanced performance of sugarcane transgenics under water limited conditions and its biosafety consideration. Sugarcane is a major source for sugar in India as well as across the world. In the world, it meets more than 70% of world sugar requirement. Sugarcane is a high water demanding tropical crop, generally grown under well irrigated conditions. Drought is one of the major important factors which limits the sustainable sugarcane production. And then this drought stress can cause potentially up to 50% yield loss to sugarcane. In India, 35% of total area has well irrigated condition, and then out of 35% of area under sugarcane, it gives 50% of total production, whereas 65% of area under sugarcane requires irrigation or requires complete irrigation or partial irrigation, which contributes 50% of cane production. However, the real situation, this 35% of total area with optimum irrigation facilities, the area is coming down. The percentage of area is coming down due to the erratic monsoon, global warming, and then over exploitation of underground water, many things, many factors. And then simultaneously, the 65% area which requires partial or supplemental irrigation, the area increases year after year. This figure, the sugarcane was cultivated in 5.04 million hectares during 2011 and 12. Then, as we experienced last 2 consecutive years, there was a severe drought condition. So the yield has been declined to 4.92 million hectares and 4.75 million hectares during 2016 and 2016-17 respectively. And this indicates, and then many of the sugar factories, they have closed down because of shortage of water, lack of cane availability. And then, as I said, the sugarcane is high water and demanding crop. Sugarcane requires 1000 to 1500 millimolar, whereas compared to other crops like cotton, it requires maximum of 950 millimeter, and rice is again 950 maximum, and then wheat requires 450 millimeters. And then not only the sugar requires more water, but if you compare the productivity per unit water consumed, sugarcane ranks first. It gives 4.97 kgs of biomass per cubic millimeter of water consumed. So this is the highest water productivity crop. And then the water requirement out of 1500 millimeter, the water requirement for sugarcane varies from in 4 different stages. The water requirement of sugarcane, there are 4 water limiting, water critical demanding periods. That is germination, that is 0 to 60 days, and tillering phase, 60 to 130 days, grand growth phase, 130 to 250 days, and ripening phase, 250 to 365 days. Germination, again the water requirement, it varies from soil type. If you have a coarse texture soil, you will need to irrigate 5 to 6 days once. If it is medium textured, 7 to 8 days. Fine textured, it's 8 to 10 days per germination phase. And then tillering phase, you have 6 to 7 days per course, and medium 7 to 10 days. Fine texture soils, you need to irrigate once in 12 to 15 days. Per grand growth period, we need to irrigate for coarse texture soils weekly once, medium texture soil 10 days, and fine texture soil 12 to 15 days. Ripening phase, it's once in more than 10 days or once in a week, it's sufficient. Out of this critical water demanding period, the tillering phase is considered as a most critical deciding factor for sugarcane yield, because if you get more tillers, even if you don't have water for 1 month, later on if you receive, if rain comes, the sugarcane will give higher yield. If you have a shortage in tillering phase, definitely your yield is going to be affected. Then shortage of water during tillering phase is will lead to 70% yield loss in sugarcane. This is a situation. And then moreover, the cultivation of sugarcane, usually we follow normal sugarcane planting, that is January to February planting. So when sugarcane reaches tailoring phase, the summer season coincides. Not only in tropical India consists of South India like Maharashtra, Madhya Pradesh, Karnataka, Andhra Pradesh, Tamil Nadu, and Kerala. In subtropical condition also the same. The tailoring phase coincides with the summer season, onset of summer season. This is one of the report given in the World Bank. 25 years of study in Ahmednagar, Maharashtra state. The yield has been declined from 106 tonnes to 60 tonnes per hectare. It was started 1970, and then finally they have concluded that this decline in the yield is due to the vagaries of monsoon and shortage of water. So, to meet the increasing demand of sugar and other byproducts from sugarcane, the yield and quality of sugarcane must be improved, but at the same time the loss of yield and quality due to drought and other abiotic stresses must be reduced. Presently, the world consumption is around 150 million tonnes. It is going to reach 173 million tonnes during 2025. India has the highest consumption rate, consuming 27 million tonnes, expected to consume 27 million tonnes in 2025. Now the present consumption is 24 million tonnes. So to meet the increasing demand for sugar as well as other byproducts, the quality and the yield of sugarcane must be doubled. So with that, only the hypothesis what we need to do is the development of drought-tolerant sugarcane genotypes or improve the water efficiency of genotype to the idea of water productivity. If sugarcane consumes per gram of water, and how much it is going to convert into biomass. So with that idea. And then we have the one variety. This is a variety is called CO86032. It is predominantly cultivated in tropical India. It occupies more than 1 million hectares in tropical India. This variety combines very high yield, high quality, and then everything is good except for moderately susceptible to drought. So, and also it is not suitable for low-lying conditions, water-logging conditions. So, presently this variety occupies 80% in Tamil Nadu, Karnataka 50%, Maharashtra 50%, Andhra Pradesh 40 to 50%. Other states combinedly it occupies more than 10%. So, to improve the productive water per drop per water productivity. So we have done a preliminary study with our cultivated variety and our wild type, and then we have compared the root morphology and its biomass characterization, and then we have observed that this wild type Erianthus aruminaceus so produces a lot of root biomass, and then it goes deeper in soil and gives a very good biomass compared to the commercial variety. The right side one is our commercial variety and the left side one is the Erianthus aruminaceus wild type. So we thought we'll try to exploit these parameters and incorporate into cultivated species. We have isolated a gene, it's called expansin gene, expansin 1. It's a gene family. It has 4 different families: expansin 1, expansin, alpha expansin, beta expansin, expansin like alpha, A, expansin like B. And then for this purpose, for this study, we have isolated expansin 1 from Erianthus aruminaceus. And then because we focus this particular variety, because this expansin, I will say in the next slide, we have observed that this expression of this gene is different, highly upregulated on drought conditions in Erianthus compared to the commercial type. And then we have used the port UB. This also again the promoter isolated from plant source, that is wild rice, Portesia corcreta. So we have focused because per cell wall is a distinct feature of plant, and its major function is to give or determine the cell structure or provide a tensile strength and protection against the mechanical and osmotic stresses. For a plant cell to grow, it must be continuously able to modify and expand its cell wall. Expansins are cell wall loosening proteins that regulate cell wall expansion. It is also involved in cell enlargement, also other development process in which cell wall modification occurs. Studies reported that expansions involved in all biological processes related to plant growth and development, including seed germination, stem elongation, stomatal opening, and xylem development, and root system architecture. So, because of this involves in cell development, cell expansion, and growth, elongation of stem, we thought whether it will play important role in changing root architecture in the commercial variety. So with that, expansion also involves in diabetic stress tolerance, but the mode of action of expansion is still not clear. So we try to put this gene in sugarcane to develop, to study whether this gene plays important role in root elongation or expansion of root cells to improve, to perform this, make the commercial clone to perform better under water limited conditions. So we have isolated the expansion gene of alpha expansion gene, the size of 1330 base pairs. This has 2 introns and 2 exons, and then we prepared a gene constructs, and then we followed the well standardized agrobacterium mediated transformation method available in our laboratory. And then this we have produced 18 transgenic events and confirmed with PCR amplifications. And then further we have followed the gravimetric method of imposing drought stress. This is how we have followed for drought screening. And then initially we have screened all these events at vegetative stage 1. The physiological parameters screened for this drought screening is relative water content, photosynthetic efficiency, chlorophyll content, cell membrane injury. So the gravimetric method of screening, the robust method was discussed with Professor Uday Kumar sir and physiology team from US Bangalore. So this is a stand-up for these 18 events. This is a tillering phase after 90 days of planting. So after 10 days of water stress, this plant reached the leaf drying, and some of them are better, shown a very good result. And then the relative water content of expansion plants, transgenic plants compared to the control plant. And also we have only the transgenic events without expansion gene. And another one is untransformed control event. And then total out of 18 events, some of them showed very good relative water retention capacity. There is a photosynthetic efficiency in the expansion transgenic events. And then chlorophyll content of the transgenic events compared to control. Cell membrane injury. So we have screened these events in V0 generation, and then we have got a very good result. During these transgenic events, during normal irrigated condition, there is no difference in much difference in relative water content between the control plants as well as the transgenic plants. However, after 10 days of drought, relative water content of transgenic events is almost 10 to 20% more than the untransformed control. This experiment showed that overexpression of expansion gene helps in enhanced drought tolerance. Once the plant is normally irrigated, it is seen that expansion gene in transformed plants were able to recover much faster than the untransformed control plants. It indicates that after exposing the plants to stress, and then once if we irrigate after the stress, the recovery of the transgenic plants much faster than the control plants. But the study is not complete because we targeted this gene for root study. This is a V1 generation. We are planning to perform the root phenotype in V2 generations. This is a transgenic part, and then as far as the biosafety issues, sugarcane is vegetatively propagated crop. Generally, stem cuttings are used for the propagating of sugarcane. But sexual sheets are not used for commercial cultivation. It's a minute, and then it is only used for sexual sheets are only used for breeding purpose, varietal development program. Sugarcane does not flower in many parts of the country. Flower means, you may observe the flowering in many parts of the country, but the seed set happens in ideal conditions in Coimbatore conditions. There is no report of pollen transfer from cultivated sugarcane to the wild relative of sugarcane. Hence, there is no chance of transient integration to the wild Sugarcane species, because we try to develop intergenic hybrids between the commercial cultivar and the wild type. But we have a lot, literally we hardly we produce 1 or 2 intergenic hybrids. Then subsequently we have a lot of problem in maintenance, and then we don't get flowering in intergenic hybrids. So the pollen transfer is not a serious problem between the cultivated and sugarcane varieties. And weediness is in sugarcane is not observed, as it is a vegetatively propagated plant and requires ideal condition for establishment and growth. And also, even if it flowers, and then there is a chance that we can cut the cane before flowering. That is possible. Then regarding the consumable part, sugar is a purified product, hence very little chance for presence of transgenic products in there, because this when we process the sugarcane juice to get the sugar, we expose this sugarcane juice to more than 200 degrees Celsius, 250 degrees Celsius to get the crystal form of sugar. So there is a little chance for presence of transgenic product. For production, yeah, it's for everything, jaggery or sugar, it's juice is processed to get the sugar. And then moreover, the development of crop specific regulation would help in reaching the technology to the farming community. This study is partly supported by DBT, ICAR extramural project, and then infrastructure from Sugarcane Breeding Institute. Thank you, sir.
Thank you very much, Dr. Chinnaswamy, for your nice presentation. I think you are in time. So we would have a panel discussion after this all the presentations are over. So thank you very much.
These events in V0 generation, and then we have got a very good result. During these transgenic events, during normal irrigated condition, there is no difference in much difference in relative water content between the control plants as well as the transgenic plants. However, after 10 days of drought, relative water content of transgenic events is almost 10 to 20% more than the untransformed control. These experiments showed that overexpression of expansion gene helps in enhanced drought tolerance. Once the plant is normally irrigated, it is seen that expansion gene in transformed plants were able to recover much faster than the untransformed control plants. It indicates that after exposing the plants to stress, and then once if we irrigate after the stress, the recovery of the transgenic plants much faster than the control plants. But the study is not complete because we targeted this gene for root study. This is a V1 generation. We are planning to perform the root phenotype in V2 generations. This is a transgenic part, and then as far as the biosafety issues, sugarcane is a vegetatively propagated crop. Generally, stem cuttings are used for the propagating of sugarcane. But sexual sheets are not used for commercial cultivation. It's a minute, and then it is only used for sexual sheets are only used for breeding purpose, varietal development program. Sugarcane does not flower in many parts of the country. Flower means, yeah, you may observe the flowering in many parts of the country, but the seed set happens in ideal conditions in Coimbatore conditions. There is no report of pollen transfer from cultivated sugarcane to the wild relative of sugarcane. Hence, there is no chance of transient integration to the wild Sugarcane species, because we try to develop intergenic hybrids between the commercial cultivar and the wild type. But we have a lot, literally we hardly we produce 1 or 2 intergenic hybrids. Then subsequently we have a lot of problem in maintenance, and then we don't get flowering in intergenic hybrids. So the pollen transfer is not a serious problem between the cultivated and sugarcane varieties. And weediness is in sugarcane is not observed, as it is a vegetatively propagated plant and requires ideal condition for establishment and growth. And also, even if it flowers, and then there is a chance that we can cut the cane before flowering. That is possible. Then regarding the consumable part, sugar is a purified product, hence very little chance for presence of transgenic products in there, because this when we process the sugarcane juice to get the sugar, we expose this sugarcane juice to more than 200 degrees Celsius, 250 degrees Celsius to get the crystal form of sugar. So there is a little chance for presence of transgenic product. For production, yeah, it's for everything, jaggery or sugar, it's juice is processed to get the sugar. And then moreover, the development of crop specific regulation would help in reaching the technology to the farming community. This study is partly supported by DBT, ICAR extramural project, and then infrastructure from Sugarcane Breeding Institute. Thank you, sir.